Recent developments in epidemiological forecasting have brought a renewed focus to hantavirus, a group of rodent-borne pathogens that remain a significant concern for global public health authorities. Mathematical models are now being used to predict how these viruses might propagate in unconventional environments, specifically looking at the dynamics of spread that could extend beyond the contained ecosystems of cruise ships into broader terrestrial populations. To understand the gravity of these projections, one must look at the foundational nature of hantavirus. These are not new threats. The virus gained significant international attention in the early 1990s following an outbreak in the American Southwest, which led to the identification of the sin nombre virus. Since then, health agencies have categorized hantaviruses into two primary clinical manifestations: hantavirus pulmonary syndrome, which is more common in the Americas, and hemorrhagic fever with renal syndrome, which is frequently reported across Europe and Asia. The core of the current concern lies in the virus's transmission mechanism. Unlike many respiratory viruses that pass directly from person to person, hantavirus is a zoonotic disease. It is primarily carried by specific rodent species, such as the deer mouse, the white-footed mouse, and various species of rats. Humans typically become infected through the inhalation of aerosolized particles from rodent excreta, including urine, droppings, and saliva. The mention of cruise ships in recent modeling data highlights a unique risk factor: the intersection of high-density human habitation with global transit logistics. If a rodent reservoir is introduced into a ship's storage or ventilation systems, the closed environment can facilitate the rapid distribution of viral particles. The current mathematical models are designed to simulate these scenarios, factoring in air exchange rates, the movement of passengers between decks, and the potential for the virus to be carried into port cities upon disembarkation. This framing is essential for viewers to understand why health editors and scientists are prioritizing this data. It is not merely about a single vessel or a localized incident, but about the systemic vulnerabilities in our global travel infrastructure that could allow a rare but high-mortality pathogen to find new pathways into the general population. While the risk of a widespread hantavirus pandemic remains low compared to highly contagious respiratory viruses, the severity of the disease, with mortality rates often exceeding 30% for the pulmonary form, necessitates this high-level mathematical scrutiny and preemptive reporting. The core developments in hantavirus surveillance are currently concentrated within specialized biocontainment laboratories and government health agencies that monitor international trade and travel. According to the latest analysis of these mathematical models, researchers are examining how the virus might move through supply chains and port facilities, a concept often referred to as spatiotemporal spread. In the laboratory setting, the study of hantavirus requires BSL-3 or higher certification due to the risks associated with aerosol transmission. Scientists at institutions like the Centers for Disease Control and Prevention and the European Center for Disease Prevention and Control utilize advanced genomic sequencing to track the evolution of different viral strains. This testing is critical because the virus is notoriously difficult to culture in a traditional lab environment, often requiring reverse transcription polymerase chain reaction, or RT-PCR, to identify viral RNA in clinical samples. The current reporting highlights a tension between the theoretical projections of mathematical models and the verified data from field hospitals and clinics. While the models may suggest a terrifying, irritating potential for spread, official statements from health departments emphasize that confirmed human cases remain sporadic and are usually linked to direct environmental exposure rather than large-scale outbreaks. However, the logistics of the shipping industry introduce a complicating variable. Large vessels, such as the cruise ships mentioned in the source material, often dock at ports where local rodent populations may carry different strains of the virus. If these rodents gain access to cargo or shipboard food stores, they can establish a colony that travels across oceans. The mathematical models are essentially stress testing the maritime industry's sanitation protocols, such as the Vessel Sanitation Program, to see if current rodent exclusion measures are sufficient to prevent a ship-to-shore transmission event. Testing centers in major port cities are increasingly being equipped with rapid diagnostic kits to screen individuals who present with unexplained respiratory distress after travel. Furthermore, the role of international shipping and the movement of shipping containers is being scrutinized, as these containers can inadvertently house rodents for weeks at a time. The scientific community remains focused on the known unknowns, such as how changes in global shipping volumes might correlate with an increased frequency of rodent-human interactions. This data-driven approach is designed to replace speculation with actionable intelligence, allowing agencies to direct resources toward the most vulnerable transit hubs identified by the models. Prevention and public guidance remain the most effective tools in managing the risk of hantavirus, regardless of what mathematical models predict about its potential spread. Public health officials consistently advise that the most critical step is the elimination of rodent habitats in and around human dwellings and places of work. This includes sealing entry points as small as a quarter-inch, using metal flashing or heavy-duty mesh, and ensuring that food sources, including pet food and birdseed, are stored in rodent-proof containers. For those tasked with cleaning areas where rodents have been present, the guidance is specific and non-negotiable. Never sweep or vacuum droppings, as this action is exactly what aerosolizes the virus, making it breathable. Instead, the area should be thoroughly wetted with a disinfectant or a mixture of bleach and water, allowed to soak for several minutes, and then cleaned with paper towels while wearing gloves, and in some cases, a high-efficiency respirator. In the context of the travel and shipping industries mentioned in reports, these prevention protocols are being scaled up to include more frequent inspections of cargo holds and passenger quarters. Surveillance also extends to the One Health approach, which monitors the health of rodent populations in the wild. Scientists know that environmental factors, such as unusually heavy rainfall or changes in food availability, can lead to rodent outbreaks, which historically precede spikes in human hantavirus cases. The limits of our current knowledge are most apparent in the lack of a specialized vaccine or highly effective antiviral treatment for hantavirus. While some countries have developed vaccines for specific regional strains, there is no globally available, broad-spectrum immunization. This reality places a heavy burden on early detection and supportive hospital care, including the use of extracorporeal membrane oxygenation, or ECMO, for severe respiratory cases. The mathematical models serve as that our understanding of viral dynamics must constantly evolve to keep pace with a changing world. While the language of terrifying spread may capture headlines, the actual work of public health involves the calm, methodical application of science to reduce risk. As we continue to monitor the data from laboratories and port authorities, the goal remains clear: to stay ahead of the curve through rigorous surveillance and transparent reporting. We will continue to follow these mathematical projections and provide updates as more verified data becomes available from official health channels. For more in-depth reporting on global public health and emerging viral threats, you may wish to subscribe to our updates. The core of recent hantavirus reporting centers on the integration of predictive mathematical modeling with established laboratory surveillance. As these models suggest a potential for spread beyond the isolated environments of cruise ships, global health agencies are re-evaluating the intersection of zoonotic disease and international logistics. Within the laboratory setting, hantavirus presents unique challenges that differentiate it from more common respiratory pathogens. It is classified as a biosafety Level 3 agent, meaning that any diagnostic or research work must be conducted in high-containment facilities equipped with specialized ventilation and strict personal protective equipment protocols. The primary method for identifying the virus remains the use of reverse transcription polymerase chain reaction, or RT-PCR, which allows technicians to detect viral RNA in blood or tissue samples. Because the virus does not typically grow well in standard cell cultures, genomic sequencing has become the gold standard for tracking how different strains, such as the Andes or Sin Nombre variants, move across geographical boundaries. These core developments are critical because the terrifying projections of the mathematical models are often based on the assumption that the virus could find new pathways through human infrastructure. The mention of cruise ships in recent data highlights a specific logistical vulnerability. Modern maritime vessels are essentially self-contained cities with complex HVAC systems and large-scale food storage areas. If a rodent reservoir is introduced into these systems, the mathematical models predict that aerosolized viral particles could be distributed across multiple decks through centralized air handling units. This creates a scenario where exposure is not limited to those in direct contact with rodents, but potentially anyone sharing the vessel's atmospheric loop. Current developments also focus on the diagnostic timeline, which remains a significant hurdle for public health officials. The incubation period for hantavirus can range from several days to several weeks, making port-of-entry screening exceptionally difficult. By the time an individual develops the hallmark symptoms of hantavirus pulmonary syndrome, which often begin with fever, muscle aches, and fatigue before progressing rapidly to severe respiratory distress, they may have already traveled thousands of miles from the point of initial exposure. To counter this, agencies like the World Health Organization and the Centers for Disease Control and Prevention maintain a network of sentinel surveillance sites at major international hubs. These clinics are trained to look for specific clinical indicators, such as a sudden drop in platelet counts combined with the presence of immunoblasts in the blood, which can serve as an early warning sign before a full viral panel is completed. The tension between the predictive models and verified information often lies in the frequency of spillover events. While the models may indicate a high probability of transmission in dense settings, verified data confirms that hantavirus remains a relatively rare disease in humans. The goal of current hospital and lab coordination is to ensure that if the model's predictions regarding a wider spread are realized, the medical infrastructure is prepared for a sudden influx of patients requiring advanced pulmonary support. This includes the deployment of rapid diagnostic kits to maritime medical officers and the hardening of shipping logistics to prevent the transport of infected rodents in standardized shipping containers. By contrasting the theoretical spread with the practical limits of viral biology, editors and health officials aim to provide a realistic assessment of the threat without succumbing to the sensationalism often found in unverified reports. The focus remains on the hard data, lab-confirmed cases, the efficacy of current diagnostic tools, and the physical barriers required to break the chain of transmission between the rodent reservoir and the human population. Prevention and proactive surveillance remain the most effective tools for mitigating the risks highlighted by recent mathematical models. While the language of these models often suggests a terrifying potential for spread, public health guidance is centered on practical, evidence-based interventions that break the chain of transmission at the source. The interaction between humans and rodent reservoirs. The primary directive from health authorities is the rigorous management of one's immediate environment. This begins with rodent-proofing structures, a process that requires identifying and sealing every possible entry point. Because certain rodent species, such as the deer mouse or the rice rat, can squeeze through openings as small as a quarter inch, officials recommend using durable materials like steel wool, metal flashing, or heavy-duty caulk. Beyond physical barriers, the elimination of attractants is vital. This involves securing food sources, including pet food and birdseed, in thick plastic or metal containers with tight-fitting lids, and ensuring that trash is disposed of in a manner that does not invite nesting. In agricultural or rural settings, the management of woodpiles and debris near the home is equally critical, as these provide the ideal habitat for the carriers of the virus. A significant portion of public guidance is dedicated to the safe cleaning of areas where rodents have been active, as improper cleaning is one of the most common ways the virus becomes aerosolized. Public health officials stress a no-sweep, no-vacuum policy for rodent droppings, urine, or nesting materials. When these materials are disturbed by dry cleaning methods, the viral particles are launched into the air, where they can be easily inhaled. The accepted protocol requires saturating the contaminated area with a disinfectant, specifically a mixture of 1 part bleach to 9 parts water, or a commercially available virucidal cleaner. This solution must sit for at least 5 minutes to ensure the virus is deactivated before it is wiped up with paper towels or rags while wearing rubber or latex gloves. In high-risk scenarios, such as cleaning out long-closed cabins or sheds, the use of an N95 or higher-rated respirator is strongly advised to provide a physical barrier against potential aerosols. These practical takeaways are the front line of defense, independent of the broader geographical spread predicted by statistical simulations. On a global scale, surveillance has shifted toward the One Health model, which recognizes that human health is inextricably linked to animal health and the shared environment. This involves multidisciplinary teams of ecologists, veterinarians, and epidemiologists who monitor rodent populations for population explosions. Historical data shows that environmental triggers, such as heavy rainfall following a period of drought, can lead to an abundance of seeds and insects, fueling a rapid increase in rodent numbers. By tracking these ecological shifts, authorities can issue localized warnings before a single human case is even identified. However, there are honest limits to our current knowledge. Science has yet to produce a widely available vaccine or a specific antiviral treatment for hantavirus infections. Medical intervention remains largely supportive, focused on maintaining oxygen levels and managing fluid balance in intensive care units. This lack of a silver bullet cure is why mathematical modeling is so heavily emphasized. If we cannot easily treat the disease, we must be exceptional at predicting and preventing its occurrence. The ethical responsibility of public health reporting lies in balancing the sobering data of mathematical projections with the reality of current risk levels. While models help agencies prepare for worst-case scenarios, such as the spread beyond cruise ships into terrestrial transit hubs, they should not be a source of undue panic. The goal of this reporting is to provide the public with the technical literacy needed to understand these threats without the distortion of sensationalism. There is an inherent uncertainty in any model, as they rely on variables that can change with new laboratory findings or shifts in human behavior. As we navigate these complex epidemiological landscapes, the commitment of the scientific community remains the same: to observe,